Always come back to the big picture. What is the goal of vaccination? It's to generate and sustain long-term memory T cells and B cells to a specific pathogen. So over here I've drawn a little graph that's also in your PowerPoint. So we think of this axis as the pathogen-specific adaptive immune response, and then down here is time. And at our day 0 we have our vaccination, and the immune response has to go through the steps of innate immunity, initiating the adaptive immunity, and then we get the B cells making antibody, the CTLs killing cells, and that process can take 2 to 4 weeks, so a very long time. So that's when you feel sick and you're contagious. So I've drawn this little squiggly line to represent that just some amount of time has passed. You've got this long-term memory response. So really important, this immune response, if it's effective, is never going back down to 0. You always have just a little population of adaptive immune cells. And then time passes. This is the great thing about vaccination. Once you get your first natural infection, well, your adaptive immune response is initiated really fast, like within 1 to 4 days. So that's before you feel sick and before you can pass it on. So this is really, really great. What we're going to get into is one reason why some vaccines are more efficacious than others is that the current vaccines that are licensed today, so those are the ones that are on the market, they actually are really great at eliciting long-term memory B cells, and so your memory antibody response. They are not great at keeping going a memory T cell response. So those are the helper T cells and the killer T cells. We're not exactly sure why. There's some hypothesis that we have some reading about, but we don't really have all the answers as to why. But it's really important to remember that licensed vaccines today are great at eliciting a robust long-term memory B cell response so that they can produce antibodies really fast. But in class, I hope you remember that not all pathogens are effectively eliminated by a B cell response. So if you remember, if all of those pathogens that infect intracellularly and can hang out inside of a cell for a long time, they're not great at being eliminated by antibodies. The pathogens that are changing their shape all the time, so that's called antigenic variation. Those are also very difficult to be controlled by antibodies because they're mutating all the time. So the most effective vaccines out there are ones that are against pathogens that can be eliminated by a B cell response. So measles is an example, polio is another example. We have the toxin from diphtheria, pertussis, and tetanus, so that's very effective. So all the ones that we think about that are effective vaccines, it's because they can be wiped out by the antibody response. In your vaccine science PowerPoint on slide where did it go? Yeah. So in your PowerPoint on slide 47, that's where we have a really nice graph that I highly suggest you get acquainted with that shows all of the vaccines that are highly effective and that they require an antibody response to be eliminated. And other vaccines that we have, like the malaria vaccine that's in clinical trials, the HIV clinical trial vaccine, these really require a T cell response to get rid of them effectively. So they're not as efficacious. Now we're working on newer vaccines to target the T cell response, but these are all in clinical phases. It takes a lot of money and a lot of years of research to get through all of the safety trials and efficacy trials. So that's why we really need scientific research support in terms of congressional funding to keep these projects going so we can get better and better vaccines out there.